I have to tell you, it's with a lot of anticipation that I get to introduce our speaker this morning. It's been a long road getting him here, and we got him. So Dr. Bill Bengston, he's a PhD in biology. I was really just blown away by the fact that he is a professor of research methods and statistics, and he's into intentional healing, which is what his topic is going to be today. He is president of the Society for Scientific Exploration, and that's something I'm sure that he'll enlighten us more about if we get a chance. This is an international group of scientists who conduct investigations into scientific anomalies. It's fascinating, the kind of things that they look into and the vast differences of professionals that are in that organization. It sounds really neat. He was talking about talking about it to me last night. The main interesting thing about what Dr. Bengston has been into is using intention to heal cancer in incurable cases in mice. So laboratory mice with incurable cancer, double-blind, placebo-controlled studies, healed using his intentional methods. And it's just fascinating to me. Be prepared to have your mind blown. And with that, I'll bring out Bill. Thank you.
No pressure there. If your mind is blown, we can get out of here early and just move on. This is going to be a 2-hour intro to some of my stuff. Is this working okay? It's not working? Okay, it's a 2-hour intro. I couldn't figure out how to give you even the basic plot in an hour. So we're going to go 50 minutes, I guess it is, take a break, and those of you who are crazy enough to come back will hear the second half and all of that. But this is going to be the main intro. And if you're seriously masochistic, I'm going to be prattling all day. Maybe I won't even stop tonight, who knows. But this next couple hours will be an intro to some of my stuff, and it will be focused on cancer, as Doug said, although we have enormous amounts of data that go to other conditions, and we can talk about that. The next talk that's a segment will be on placebo responses in animals. And I'm going to suggest to you, for those who are more methodologically inclined, that placebo effects are not suggestion. It's not a psychological phenomenon, but is an actual condition of resonant bonding, and it's a physical condition that can be measured. I'm going to suggest it's more of a sociological phenomenon rather than a psychological phenomenon. And we're going to look at some of the patterns of placebos in people and get rid of that pretty quick. And then I'm going to talk about how I've induced placebo responses in animals. I also have produced placebo responses in cell cultures. And now for the kicker of all kickers, I've induced placebo responses in placebos themselves. I don't know where to go from here, because I think that, I got nothing after that. But so that will be the third talk. This being 1 and 2, we'll then talk about, for those really crazy and coming back, subjective states of consciousness. And this is the question of, when we're looking at hocus pocus healing, I tend to make fun of the stuff, when we're doing hocus pocus non-traditional healing, what is the state of mind of the healer, what is the state of mind of the healee, and what happens between them? And I have EEG data, functional MRI data out I'm a data freak. So that will be the point of number 4. And number 5, which will be later in the afternoon, can healing with intent be captured and reproduced without the healer? And this is the project that I'm working on mostly now. I got several of them going. But the general idea is, can we make healing scalable? There's only so many people with so many hands and so much intention. Can I capture it? Make it scalable, and how do we capture it? And so we'll look at some of the data on capturing it biologically, and it turns out biology can remember and store healing information, and stuff can remember and store healing information. And right now, we're working on even the possibility of making a downloadable app that'll reproduce the healing without the healer. And so I've got 84 cancer genes examined. We're starting to get a mechanism, to understand the mechanism by which cancer is cured. Those who want to, this will be about primarily data, and there'll be a break between 5 and 6, if I'm understanding the program correctly. And we've captured and we've played we have a recording that we're studying in the lab. And for those who can't get enough, during the break, because it's not going to be technically part of the program, but during the break, we're going to play the recording of the captured healing, and so you can experience it for yourself. If you have sick dogs and cats, we can fix them, we'll bring them back. And then finally, number 6, and this will be, I have no idea what time this will be, probably Wednesday. We'll talk about how to do it. And the general idea here is I don't have any proprietary information. I don't have little secret handshakes and techniques. My stuff is published. It's public. People all around the world are doing it. Vets love this stuff. Physicians, MDs, not so much, because physicians aren't allowed to cure. It turns out you get all bent out of shape if you fix a person. I call them uprights. If you fix a dog or a cat or a horse or whatever, everybody's happy. And so vets can just go out there, and I get emails from vets saying, hey, finally I can cure the dogs and the cats, and I never could fix the cancer before. Well, if you want to know how to fix it, I'll show you how to fix it. And so this will be a discussion. It's not going to be a training session that's going to go on. I'm going to give you the generic plot and how to find out how to do the whole thing if you'd like. And so this is the arc of the day. Maybe Wednesday's too early. It could be Thursday by the time we get down there. But we'll give it our best shot. And we're a reasonably small group. If you have a burning desire to ask a question, ask a question. I'll probably duck it. Now, the general idea where I come from, and I don't mean New York, where I come from, a long, long time ago, in a place far, far away, I was thinking of writing scripts for movies, but a guy came along and fixed me of a bad back. I found that to be rude. I'm not a believer in this stuff, and this dope comes along. I'm in pain, chronic pain for years. I had to give up a swimming scholarship. Chronic pain for years, and you learn to live with pain and all this kind of thing. You just move on. You're going to go through life in pain. And this loser comes along, puts his hands on my back, and I haven't had a pain since. And I find that rude. I'm not a believer. I'm still not a believer. 100 years later, I test. I don't believe. I look for evidence. I don't start with a conclusion. I keep poking and poking and poking and poking and poking. So I don't come from a position of belief, but this loser comes along, puts his hands on my back, pain goes away. I haven't had a pain since, so I got 2 possibilities at least. One, most common, let's walk away and pretend it never happened because it's too scary. 2, figure out what in heaven's name just happened. For better or for worse, I went down the second road, and I've been stuck in labs and people and all this kind of thing ever since. So I've been doing this thing for decade after decade. I started with the uprights because I am an upright, you probably can tell, and so I'm an upright, and this guy puts his hands on and the pain goes away. And then I said, well, that's crazy. Here, let me put your hands on something else and see what happens. And let's put the hands on something else and see what happens. So I started dragging around his hands and say, here, give me some hands, put them over here, and do this. And we came into some reasonably interesting patterns. Some things didn't respond so well. Some things responded very well. And it was curious. One of the things that responded very well was cancer. And it turns out the more aggressive the cancer, the easier it is to fix. And so we did a couple of 100 cancer patients, the uprights, and then I realized that I wasn't learning anything. All I was getting was, person comes in like me, you're in pain, you got a tumor, you got whatever you got, and they get a series of treatments. And it's not, magic, it's not snap your fingers and everything goes away. But the cancer was going away, the cancer was going away, the cancer was going away, and what do you do with that? And I wanted to understand what's below it. What is the phenomenon that we're watching? And so I said, we got to get away from uprights. and get into the lab, and let's do this under seriously controlled conditions and see what happens. And under seriously controlled conditions, the same stuff happens, but now we can walk away with a little bit more of an in-depth understanding. If someone comes to me and they say, I've got condition X, it's an algebraic thing. So they have condition X, and they come in and they've just had a glass of water. Well, was it the water that fixed them? Was it the grapefruit they ate? You see I'm a skeptic? Was it the grapefruit they didn't eat? Was it the exercise? They stopped exercising. They rode a bike. I have no idea. So it's hard with uprights because you can't control the environment and you can't control all the variables. I needed to go into the lab, and let's do this under seriously controlled conditions where there's no real viable counter-hypotheses. If it works, we'll know exactly what's going on. It's not just, here's an anecdote. If I come in here and I say to you, I used to be in pain all the time, now I'm not in pain at all, you got nothing. You believe me? You don't believe me? It's an anecdote. Maybe I forgot about the pain. Maybe I forgot about not having pain. Maybe I'm exaggerating this. Maybe it turned into a fish story. Maybe it, like that. And so I needed to go into the lab to do this under very, very controlled conditions. So I've been doing this for 115 years. And in terms of major goals, I've been trying to figure out the parameters of healing. And by that I mean, what are the things that make it work well? What are the things that make it work not so well? So for example, I'm just going to make stuff up. Does belief matter? Does it matter whether the healer believes or not? Does it matter whether the healee believes or not? And it turns out, I think it does a little bit. I'm speculating here, though. I think all other things being equal, that's a statistical cop-out, all other things being equal, I think belief retards healing. Just a little bit. So you don't want a believer. Someone comes in and goes, I believe, and I go, I'm scared of you, and I run out of the room. It's belief is not the important, and we're going to see this in a later talk this afternoon, where we're going to see that healing autonomically kicks in, even without conscious awareness. And I got this controlled in 2 medical schools. That even the awareness of the healer doesn't matter. And if you insert what I pejoratively call the P-brain into the equation, you retard healing. So the attention of the healer or the attention of the healee only detracts. And of course, I've got a title here, Healing with Intention. So it's just, it should have been Healing Without Intention, yeah.
What's the definition of intention?
Intention, yeah, okay, that's good. The question is, what's the definition of intention? The answer is pretty simple, I don't know. But the intention might not be a good word. I mean, thanks. It could be focus, attention, paying too much close attention so that the conscious mind is focused. I don't think the conscious mind being focused on anything ever helps. All mastery of any skill requires mindlessness. You begin with mindfulness, and let's just say walking. You begin with mindfulness and you're paying attention, when you're first starting to walk, but then you've all transitioned to mindless walking. You don't remember walking. Why? If you go back and you mindfully walk, you look klutzy because you're paying attention. You learn a sport. You drill, you practice, you drill, you practice, but then you got to let go. Healing is also letting go, in my opinion. No, in my evidence. So I'm looking at the parameters of healing. Well, a personal thing that, do you believe or not? I don't think belief helps at all. How about distance? So I got a dog here, and I put my hands around the dog, and I do whatever my hocus pocus method is, or I have a dog in Australia. Does it matter? Turns out, no. And so we have experiments done at 2 inches, replicated at 2,000 miles, exact same results. Does dose matter? How much do you got to treat? How often do you got to treat? Is an hour treatment 2 half-hour treatments? Or is there a decline during the course of a treatment? If I do 2 dogs at a time, do I get a different response than if I do 5 dogs at a time? You get the idea. So dose, distance, and we're going to talk about all this kind of stuff during the course of the day. So I'm just kind of interested in the parameters of healing. What are the physiological correlates of healing? And that's one of the talks later. And that will be, I mean, I've done mostly brain research, so I'm looking at brains. Turns out brains can go into a phase lock at a distance. Reasonably interesting. So I got EEG, EKG, a bunch of Gs. And so I'm interested in the physiological correlates of healing. Teaching efficacy is interesting. That'll be our last topic for the day. Can you teach this stuff? Turns out it's a pretty difficult problem to solve. But people all around the world are using my stuff and fixing stuff. It's interesting. The scientific mother lode is the development of a theoretical model. I'm not close. And so I've been doing this for 130 years. I'm not close to a theoretical model that understands how it works. I can make it occur. I can give you the conditions under which it occurs. But really what's going on when you do healing, still past my pay grade. And so I figure since I've been doing this for 130 years and I'm probably not going to do it much more than another 100 or so, I ought to build something practical. And this is what we'll play with in the 5th talk. Can we capture and reproduce healing without the healer? Can we make this a scalable proposition? Can we turn, for example, the cancer cure here into a cancer vaccine? And it turns out, just looking ahead, we can. So we know enough now to make a cancer vaccine. Whether we're able to use it, at least on the uprights, there's a 3-letter word. It's worse than a 4-letter word. It's called FDA. And the FDA is not going to let us go sticking needles into people and injecting them with this, that, and the other. But you'll see from the animal work, we're reasonably confident we can make a cancer vaccine. That's the good news. The bad news is we'd never be allowed to use it. So what do we do instead? And we'll see that we've actually captured something on a recording. We've looked at 84 cancer genes, and the data they're reasonably interesting. So this is the plot that I've been working on for 147 years. And we're going to touch on just little bits here. The first couple of talks, 1 and 2, is kind of like a quick down and dirty overview. This is an advertisement, but not really. Sometimes I write in English, not often. And this is a story. So it's once upon a time, and some idiot fixed my back, and things like that. But it's just kind of like it's cute. But if you want to learn the stuff, this is an actual training course in the energy cure. And it turns out there are people around the world that I've never met in person who are sending me reports of, I studied your dopey technique, I've been practicing my brains out. It's really a pain, incidentally. I'm practicing my brains out, and I've just fixed, and I've just fixed, and I've just fixed, and I've just fixed. This is hard on a non-believer. So I'm getting stuff from Australia, and they're saying, yeah, we just did this, or we just did that, and I'm going, I don't believe this stuff. But if you want to know how to do it, this is how to do it. If you like Once Upon a Time, this is Once Upon a Time. And at the end of this, I'll give you a whole bunch of different ways to learn how to do this for yourself. It's not a faith-based thing. It's a practice. It's pretty interesting. The results are pretty interesting if you decide you want to do it. Again, don't do uprights. Somebody will come out and get you. So sometimes I write in English, but most of the time I write stuff nobody reads. This is for journals and more academic kinds of stuff, so crossing disciplinary boundaries, going beyond meta-analysis. I mean, nobody's going to read this crap. I wouldn't read it if I didn't write it. So these are journals, these are journals, these are journals, these are journals, these are journals. I write stuff that nobody reads. And so we have a little inbred group of people who share and doing studies and things along those lines. So I'm going to give you, for now, this talk and the next, 5 propositions. And we probably won't get past the first one in the first hour. And these are going to be much shorter. We may not even get past the first one in the first hour. But first proposition, we can cure cancer. What I don't know is about the energy healing part. And so I've got energy healing in quotes. I'm reasonably confident, and probably wrong. I'm wrong about almost everything. I'm reasonably confident, but probably wrong, that there is no energy in energy healing. Now, the book I just showed you is called The Energy Cure. And I went to, when the publisher showed me the cover, and I said, it's The Energy Cure, and I said, but I don't think there's an energy. And they said, thanks for your input. And that's as far as I got. You want us to do the book or not? You know, we're going to call it The Energy Cure because everybody understands that. I think it's actually information. And I'm trying to isolate the information, and it turns out to be a pretty difficult problem. And I suspect that what people call energy healing is not energy healing, it's an informational transfer. And I may talk about that sometime down the road. So if I do a cage of mice at 2 inches and then do it at 2,000 miles, or I do it in Australia, energy? I mean, there's a pretty simple formula to understand the diminishment of energy over distance. Healing doesn't diminish. How can it be an energy? Yeah.
My understanding is that information is energy.
Some people think it's fungible, but energy has a particular vector. It's got a power. Whereas information can just be sitting there as a potential. And so maybe they're fungible. I don't know. I don't really know. I can't get a straight answer from anybody who's looking into this thing. I suspect that we have an information packet which is riding an energy wave, but maybe we'll get into that down the road. So the first proposition, if we take out the energy part, is we can fix cancer. And it's not a remission, it's a cure. I used to use the word remission and remitting and this and that, but I got yelled at by some colleagues, and they said, you're not remitting, you're curing. It's not diminishment of symptoms, you're fixing it. They're immune for life. You'll see that even if we re-inject mice with cancer, they're immune for life. Not only are they cured, they can't get it again, which is reasonably interesting. Can't get the same, we haven't tried different cancers, yeah. We're going to suggest there's a dose response. This isn't magical things. People think of healing as magic. But there seems to be patterns of dose responses. Certain length of time required to produce a certain effect. Working on, should I do it in one dump, or should I do it in mini dumps? Should I? These are unknown, but there is a dose response out there. Healing is nonlinear. And you'll see that it's akin to a phase transition. Nothing seems to be happening, nothing seems to be happening, nothing, and then suddenly this stuff happens. It's actually very, very interesting. And I'm going to suggest to you finally at the end of the first couple of talks, healing and killing may not be compatible. And that sounds like it's facetious, but I'm not being facetious. So if you have a cancer, door number one, I'm going to kill it. You know, we say war on cancer, we do all this kind of thing, and we drop bombs on it, and we radiate it, and we do, and yada, yada. Go ahead, kill cells, have a ball. Killing is not the same as healing. Now, if you're an upright, and you have a bad diagnosis, you have a tendency to throw the kitchen sink at it. Give me this, give me this, give me this, give me this, give me this. I mean, you understand that. It's not helpful. Kill if you want to kill, heal if you want to heal, but combining the two, not so good. And we'll kick that around, yeah.
Meaning that you can't, those therapies, healing therapies won't work if they're simultaneously on killing therapy?
I can't say they won't work. I would say at the very least they're drastically diminished in efficacy. And the only clinical cases where we've had failure has been when we combine healing and killing. If you don't include killing, we don't have failure. I understand that's a crazy statement. Scream. I'm sorry, I missed it. Yeah. No. No. Yeah, no. It's almost like waves canceling each other out. And what would appear to be the case, and this is simply an anecdotal observation, is that the healing tries to heal the killing. So there's a hierarchy which doesn't come from your intention. There's a hierarchy, and healing comes from the healee. And if the healee has just been recently damaged and you offer healing, the healee will try to get rid of the damage. And so there is a hierarchical need. And we'll get into some of that stuff at some point during the day. You got to scream because I'm the only one with a mic. Yeah.
So, obviously, as holistic vets, we tend to do more healing therapies.
Yeah.
Which are, supplements and such. They wouldn't be part of that healing.
Right.
I mean, have you done some?
Yeah, supplements, anything which enhances healing seems to be fine. But if let's say we got a tumor here and then we are radiating it. Well, we're trying to kill cells. And we're hoping our target's good, we've done enough, not too much, et cetera, et cetera. But we're trying to, and radiation will kill cells. There's no question. But when you come along and you've just been killed, you're killing cells, you have a wound. You're hurting. And you come along and you offer healing, I think it goes to that first. And so, but, and we start throwing the kitchen sink.
I think her question was about other things like supplements and things like that.
Oh yeah, that's why I said supplements are fine and all those things which are enhancing, particularly something which enhances the immune system. Anything like that, as far as I'm aware, is fine. So these are going to be the 5, wow, I talk too much. We're going to get through some of number 1, and I guess when Doug says, zip it, stop talking, we'll stop, take a break, and then keep going, but I guarantee you we'll get through this within the first 2 sessions. So this is the plot. So I'm going to focus on cancer because most of my work has been on cancer, and I need to apologize to you as a holistic vet's place. Some of the research is ugly. It is ugly. There's no way to make it anything other than ugly. It's also, and some people disagree with this, I got to do it this way in some cases. When you do in vitro work and you're dealing with cell cultures and such, there is not a direct translation from in vitro to in vivo. Bodies are more complex than well plates. And so you can do this, but you also got to do that. And so you're going to see a couple of ugly pictures with this. It's ugly. There's no other way to put it. But there's no other way to get it. So what I'm going to draw on, these are in vivo cancer experiments, and this is the number of experiments in different places. So I've been on the road a bit. I've got 2 formal experiments on a mammary cancer in mice at City University of New York. 2, 1, 3, 3. We're doing work now at Brown. On a different kind of cancer, a sarcoma, I got 2 at the University of Connecticut Medical School. Oncogenic mice right down the road here in San Diego. Nude mice. I don't want you to worry. I'm not going to show any nude mice pictures here. So I don't know whether vets, like their animals clothed or unclothed. But I took it out. Margaret made me take it out. I had nude mice in there, but she said, no, they'll be offended. Extremely aggressive cancers, this at Wake Forest, and then I got, I'm not going to keep going, but there's in vitro leukemia cells, breast cancer, and we're running a whole bunch of in vitro stuff as we speak now in Providence. So I'm drawing on a bunch of data. I'm not talking about somebody who comes along and puts their hands on my back, and I'm saying, oh, it feels better. I'm talking about under seriously controlled conditions here. So the first proposition, cancer can be cured. And the quotes here are because who knows. And for cancer can be cured, I have experimental evidence and I have clinical experience. The experimental evidence is all under controlled conditions. The clinical experience is not. Someone comes in, hocus pocus, someone leaves. Was it the hocus pocus? Was it something else? Was it? Don't know. And so I make a big distinction between clinical experience and experimental evidence. On uprights, we have hundreds of clinical cases. So we're talking a database in the hundreds before we went into the lab, and we have hundreds since. I don't do clinical stuff anymore, but there are folks who've learned my technique are doing clinical stuff all over the place. So I ship them out because I'm scared of uprights. So for this, cancer can be cured. I've got evidence and experience. The only thing I'm really going to show you here is evidence. Now what I do as a model is I take something that's already thought to be well known, thought to be understood, and I introduce a single variable. That's the idea. So in this particular case, and this is just gobbledygook stuff, here we have the bottom line in bold, is that we're taking a mouse model that has been studied and studied and studied and studied. There's over, this is literal, there's over 2,000 publications on what you're about to see in the peer-reviewed literature. So everybody knows what's going to happen, and if we got any bio geeks in here and you saw this, 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 and this, you'd understand that, but the only thing that matters for us is 14 to 27 days. In over 2,000 experiments in labs all over the place, a particular kind of mouse is injected with a particular kind of cancer, and it follows a very predictable pattern. No mouse dies before 14 days after injection, and no mouse has ever made it to day 28. Now this is literal. No mouse has ever made it to day 28. The distribution looks like a perfect normal curve. The standard deviation is 3 days. And so 2/3 of the mice are dying plus or minus 3 days. And you know, and you can look up on tables, what percentage will be alive after day 15, what percentage will be alive after day 20, and you have 100% death no matter what you do to these mice. They've been fed, not fed, chemotherapied, radiated. No mouse has ever made it past 27 days. So there's a huge database on this mouse. Everybody knows it. It's done all over the world. It's the most common mouse model. There's nothing exotic here. And you follow a standard biological protocol, and then I introduce one variable, hocus pocus. What happens to the distribution? That's the question. So here come the ugly pictures. This is a non-metastatic cancer. It grows, it grows. Now this is, I mean, this is ugly. So you have here, this is the tumor. You can tell, and everybody here would be able to recognize, this is not a healthy mouse. You can't see the eyes, but the mouse isn't there. The fur is falling out. This is it's a non-metastatic. You die of malnutrition and/or the crushing of the internal organs. It's not a good way to go. But this is the pattern of this particular model. So it's done thousands and thousands of times, and the general idea is, you know, we got to make it stop. I got a note from this guy once. And so, you know, we just stop doing this crap. And so it turns out the answer to the problem is this. Now, I got a lot of pictures of a lot of people doing this stuff. I don't want to out them. I'm just going to use pictures of me in this. And sometimes I function as a healer. I'm not a healer. But I do the technique. But I normally get, in order to be healers, inexperienced faculty and students. And I go to them and I say, you believe in this healing stuff? And they go, what? Perfect. If I got somebody and they said, you believe in this healing? Oh, yeah. I say, get away from me. Believers scare me. I don't understand the basis of their belief. And so I've dealt with volunteers, students and faculty, who are inexperienced non-believers. And they function as so this is just me at Indiana University Med School, and this is about as exotic as it gets. So you stand there and you sit there and you have your hands around a cage and you do a particular technique that you don't know about yet. I'm doing it right now. It's not a distraction. It doesn't take away from anything else. It's just something that I have incorporated into living. So I'm doing that, and I'm going to come back to this when we get to subjective states later in the afternoon. I assure you I'm not meditating. I'm sitting there with my hands around a cage going, damn, this is boring. How much longer do I have to do this? I hate this. And the clock is going, tick. Tick. It's ugly out there. Now, in this particular protocol, you can see I got cage, 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 cage. An hour a day per cage. Yeah. You got to be a dope to do this, but I'm a data freak, and this is what I got to go through. So there were 6 rooms going on at the same time here in a particular building in Indiana. And so I was doing hocus pocus in one room, and then they had a student volunteer doing the same thing in another, and they had someone who knew my technique in this room, and someone who didn't know my technique in that room, and someone else waving their hands, and we do a lot of different stuff. We also have multiple experiments going on, which I may allude to. This geomagnetic probe, and we're measuring physical space in the room. There's in different places in the room, there are different probes of different orientations, and we're taking measurements of physical space. We're running random number generator. We got a lot of stuff. And so we're doing this, we're doing this, we're doing this. We got enough data just out of this run, to last us quite a bit of time. But we just take folks who do this, and it sounds exciting, so someone here comes up to me, making this up, and says, can I be part of your experiments? Be careful what you ask for, because you may sit there, tick. And I don't know if you're allowed to say nasty things because we're being recorded, so I shouldn't say shit or something like that, right? So you say, oh shit, I got to do another cage. I thought this was supposed to be fun. Or I thought this was supposed to be, it's just, oh God, it's awful. That's why we got to record it. Healing is ugly, because it's boring. Now, here's what happens when you do my hocus pocus. Here's the plot line. I assumed 142 years ago that healing would be something analogous to radiation. And if we got to mice early enough, so we get a cage. Cage is prepared by a biologist who does this, and you get the mice with cancer a couple days after injection, and you go zzz, zzz. I mean, you don't actually do that, but you go zzz, zzz, and you're doing you're zapping. Okay, so we're zapping. And I'm thinking, how should we think about this stuff? So I go zap, zap, and then the tumors start to grow. And I say, well, that's not good. It's not working. And non-believer that I am, I said, let's call it off. It was hard to set the first one up. This didn't come easy. It's a pain to do this. And so a heartless friend of mine who had been instrumental in setting this up said, we just spent 6 months trying to put this together. Do it a couple more days. And I'm going, but look, they got tumors. Just give it a couple more days. And I'm going, oh, this is dopey. So I'm going, zzz, zzz, and the tumor gets bigger. I don't think it was a direct relationship, but you get, and the thing keeps growing. And then they develop an ulceration on the tumor. And I'm jumping up and down going, folks, it's not working. Pay attention. How does belief matter? It's not working. Call it off, call it off, call it off, call it off. And my heartless friend says, 2 more days, 2 more days. Tumor ulcerates. I'm freaking out. 2 more days. I mean, by all accounts, it's not working. And then it collapses. And the mice are cured. And so this will go to a pattern which we see in uprights as well as the more interesting ones. And we see this in a lab. We see this out in the field. We see this. Healing is not some magical thing. There seem to be patterns and rules which certainly didn't correspond. My point here is that healing doesn't have anything to do with what I want. What do I want? Well, I want magic. What do I want? I want to wave my magical hands and everything goes away. What do I get? What nature throws at me. So after you do this for a while, you lose a bit of the arrogance. I thought, zzz, zzz, that would do it. No. So here's what we have. This is days subsequent to injection. does happen. That's why I could say it out loud, because it does happen. It's natural. So you see the beginning of an ulceration there. Now, if you don't have any other data, you think, well, there's something the matter here. You know, why are we doing this? It's not. Now watch the next one. Now look at the coat. If you don't know what you're doing, Exhibit A, if you don't know what you're doing and you see this, you go, well, this isn't good. But you look at the coat, and if you could see the eyes, they're clear. And the animals are running around in their cages having a good time. And I'm yelling at them going, sit quietly, you're sick. And they're fighting with each other, and they're having fun with each other, and they're doing all this other kind of stuff with each other. And then subsequent stuff, which I don't have right here, the thing collapses, covers nothing. If I do histology here, there's cancer. If I take blood, there are cells. When they're cured, there's no evidence they ever had cancer. It's reasonably interesting. Now this is a different kind. Normal progression, different kind. You can see that this is sarcoma. It's also non-metastatic, so it's easy to work with. You can see and you do the hocus pocus stuff. And one of the really interesting things, if you do it on the white mice, is that they grow arrows on their back. We don't understand why that is. If anybody here knows graphics, and they could explain what that, but here's what happens in this particular kind of cancer. In the last one, the mammary cancer in the brown mice, they all follow the same pattern. No mouse has ever been cured without getting a tumor, step one, without the tumor ulcerating, step two, and with an implosion. They vary very much in the speed with which they go through the process, but the process and the stages are universal. In this particular form of cancer, we got this big ugly looking thing. You can see the analog to the brown mice. And now sometimes you come in the next day and it's like somebody let the air out. And then of course we have the anomalous arrow on the back. And other times it goes that's pretty similar. You can see the similarities to getting the ulceration and then the thing implodes. Now you can see there's no sign. And blood tests, histology, no mouse ever has any sign of any cancer for the rest of their life. And then there's more stuff. Yeah?
I may be jumping ahead, but do you do that with tumors that are growing internally and ulcerating somewhere?
We don't know about the ulceration, but we for example, leukemia. So we're doing that by blood counts and things along those lines. So the leukemia goes away, but it doesn't go. Each kind of cancer seems to have its own pattern of getting there. And I think the timing's going to work. This is just an example of, you can do this on this is an upright. And so I'm showing you about 6 weeks apart. A breast cancer, you're looking at things in an MRI that you don't want to see. You're looking at things that you do want to see. So we've got hundreds of human cases, and they go through a particular stages, and they go through, and they go through, and they go through. We can do it by MRIs. We can do it by thermal imaging. We can do it by cancer markers. We can do it by however you got diagnosed is the same way you're going to be pronounced cured. You know, it's not believe or anything like that. Nothing spiritual is going on. This is just a natural, normal process that we seem to be able to instigate. I think I did it, Doug. Yep. Any questions for this part before we take a 10-minute break? Yeah. I don't know what the denominator is. I don't know really what happens, what percentage get better. The only failures I'm aware of in uprights are those who combine healing and killing, is so long as they stay throughout whatever it takes to get them cured. Probably too long of a winded question. Answer.
Was a necropsy performed on that mouse that died just to see if there's a concurrent issue going on?
Nothing. This was early on when the mouse died, and all we did was confirm that it was indeed cancer. And so it was just superficial histology in this, but we didn't do any crazy stuff. It died the way it's supposed to die. And there was nothing unnatural, meaning by the model and all the publications, it's supposed to do this, it's supposed to look like this, and it was doing what it's supposed to do. Well, its companion in the cage got cured. This goes, you're looking at a statistician, we call it unexplained variance, and what that really means is I have no idea what the hell is going on. Yes, unexplained variance sounds better, but it really is, I got nothing, yeah. Yep. No. These are standard. They're sitting in cages. They got feeders, coming in bulk things into the animal facilities. And I don't know exactly what was in the mice food, but there was nothing exotic going on. And so I've never done any experiments that involved trying to alter nutrition. Certainly in people's cases, and this goes to complications in trying to figure this out, you have patient one, and patient one goes on a fast, and patient 2 goes on a grapefruit diet, and patient 3 goes on and patient 4 goes on, I mean, you get the idea, and I can't control that, and this is one of the reasons I had to do it under controlled conditions. But what the mice are really eating, I don't know. Yeah, standard stuff is about, but I have no idea what that means, really. Any other questions? Yes?
How many mice were involved until now?
How many mice were involved is the question. 300 something. Yeah, this isn't like, a mouse. This is a lot of stuff in a lot of places. The largest experiment we did was with 75 mice. And this will go into some of the things I'm going to comment on here. Does the number of mice or subjects matter? And it turns out it does, but we'll see that in a second. We're good? So the goal now is to just give you a quick ditty on this is really a cure, it's not a remission. This is not magic. There's a dose response which is still being worked out and we certainly don't understand. And what I'm going to try to do on number 3 is shift your mind from the idea, there's a lot of people out there, my experience, who think of healing as it's something special, mystical, sacred, something. It's just healing. You're healing right now. This stimulates something else. Relax. But it's not magic, is the point. There are rules and regs, and people aren't looking at that because they're going, healing, and they're flopping on the floor and doing all that kind of thing. Well, it turns out, there are interesting patterns which occur. And there is a dose response. And so wouldn't it be nice if the world conformed to my wishes? Yeah, I'd like that. And I, wave my magic wand and it becomes as I want it. That's a beautiful thing. But eventually you turn 3 years old and you find it doesn't happen. It doesn't happen in healing too. Healing never works the way I want it to. Nature is very rude. And so it does its thing, and I'm trying to figure out what are the dose, what are the patterns, what are all those kinds of things, and what's the variation. And we go down to number 4, which will have tremendous, and I'm putting this up, I got a whole bunch more propositions, but 4 matters, particularly for those of you here who want to go do this. And so if you're going to go take your animals, and we've done horses, we've done pigs, we've done dogs, we've done cats, we've done, we've been out there in the vet world and we've done some experiments with all sorts of different types of critters. If you want to go do that, there's a lot of clinical implications, because particularly if people think magically, so if I'm giving a dose of whatever that is, and I say, take this 3 times a day and take this and people understand that and then they dose it out. But when you're healing, they go, magic, make it all go away, or it just doesn't work that way. And if healing occurs nonlinear, it becomes really difficult to manage on a clinical practice. So some of you may decide you want to do this and at the very least just get rid of the cancer in your animals. But it's not simple, meaning it's a bumpy ride. It's not like everything gets wonderful because you wave your magic hands. And then this also will have clinical implications because presumably, certainly for the uprights, but presumably some of your clients, some of your patients, some of your owners, I'm not sure the right terminology, may say, get rid of that tumor or do this or do that, and it's ugly out there. So cancer can be cured, not interesting anymore. I mean, it's literally not interesting. If you've got a few hundred mice and you've got a few hundred people and you've done this in 7 different labs, it's no longer an interesting question. The question that's interesting is, what's the mechanism? What are the parameters? But not, can you fix cancer? It's just not interesting. Now, other things which are equally interesting, which I'm ducking here, because Doug won't let me talk through Wednesday. Early Thursday? Okay. Other conditions respond very differently than does cancer. So again, it's not healing versus no healing. What are the rules? What are the patterns? And there's some things we don't affect. And if you're looking for a research agenda, which if you stay here long enough with me, you're going to want one. But if you are looking for a research agenda, comparative healing. So healing method X relieves pain. I'm making this up. Mine sometimes makes pain worse. Healing method X can fix warts. My method won't fix warts. And I've had people come up to me and say, any idiot can fix a wart. And I said, well, this idiot can't. And people who do warts, they do Reiki warts or they do whatever warts, and they do, and you go up to a wart, and I've even had people mock me, you just go boo, and it goes away. Well, if you're using my technique and you go boo, it mocks you. And you can treat it all day and all night. I got a $5 million reward for anybody who can do a wart. You can't sneak and do Reiki. That's cheating. But Reiki doesn't fix cancer. That's interesting. Now, between cancer and a wart, I don't have a hierarchy. You might, but I don't. It's simply a different response. So this technique fixes that, but not that. This technique fixes that, but not this. You want a research agenda, that's completely wide open. Is there a chemistry of healing? Can I combine this with that? So if I promised not to bring nude mice in here, and you got bottles that say naked on it. So if you get naked and combine that with healing, does this combine differently than something else? There's no chemistry of this stuff because people think in magical terms. The question, nutrition. If you add nutrition to these are all, it's wide open. So it's interesting out there, and the hardest thing is people go, well, this isn't possible, so would you do it one more time? No. You do it. Have a ball. I got a call the other day from some head of some big Israeli hospital saying, if you would just do the mice in our hospital, I said, get away from me. You're a loser. What if you're listening? You're a loser. I wasn't subtle. And I said, it's not interesting. You're just trying to see if you can fix cancer. It's not interesting anymore. Give me something I don't know. So give me a toy you've got there so I can get a different data point. I don't need the same crap over and over again. Enough already. Even I, as a card-carrying skeptic, get it. You can fix it. How? Why? That's what I'm working on now. So we'll go through 2 through 5, and not as long as number 1. And you already got the plot for proposition 2. Once it's cured, I mean, it's kind of redundant there, it's cured for life. And once again, I used to be thinking in terms of remission and how long and this, that, and the other thing. But this is full lifetime cure. So for this, I have an enormous amount of experimental evidence, and I also have clinical experience but the clinical experience always comes with an asterisk, meaning, I don't know. I don't know what they did. I don't know what they ate. I don't know what they didn't eat. I don't know. And a lot of people come, they're expecting magic and they run away. Well, what happens to them? I don't know. How many treatments? I don't know. 25 years later, did they ever get anything else? I don't know. Mice, I know. So after energy healing, using in quotes, to full cure, in hundreds of mice, we've never had one with a recurrence. And we re-inject them. And we've done this in multiple places. So here the mice go through, they grow the ulceration, or they grow the tumor, the thing ulcerates, it implodes, and they hang out there. And biologists can be really sneaky, and so some of them go and they re-inject them. They can't get cancer again. And this lasts their entire life. They don't live longer than normal. You know, we don't have Methuselah mice. If they're going to live 2 years, they live 2 years. They're not, you know, and then they die. But they die without cancer. 1 and 2, yes.
Did they re-inject with the same?
Same. We haven't done it with different cancer. So this is, I got a scroll of things I want to do. One of them is to re-inject with a different cancer, and that's going to have all sorts of immunological implications. I don't think they'll be immune to other kinds of cancers. Having said that, you need to understand I'm almost always wrong. If I say A, you can become wealthy by betting on B. I once went 20 years, this is literally true, 20 years without getting a single hypothesis right. And what that means is, I don't have a clue. And I got news for you. Anybody who claims to have a clue, my penny, run from them. Because if you take your ideas and you actually put them to the wall and test them rigorously, you're going to find out the things you believe probably aren't real. Certainly the things I thought these are. That's what's fun about research. You're looking at the dumbest guy you've ever seen, because I've tested everything and I got nothing to defend anymore. I understand I don't know how the world works. And it's really fun getting data points. Because then next week, literally, next week, I'm going to get a call from the lab in Providence and they're going to go, and be yelling, this is what we found. I'm going to be going flopping on the floor having no idea why. And so I go through life completely confused, and I've grown accustomed to it. So please, don't listen to me. Yes? Oh yeah, should you listen to me? No, you shouldn't. Yeah, yeah, I mean, yeah. This should be called Never Mind. Yeah, only kidding, and he's making the crap up, you know, so. So we get to the third bullet, and it's got the qualifier, as far as I know, no person ever had a recurrence. But what happens really? What happens 30 years later? I don't know. What happens with the mice? I know. And so I've got a speculative, I actually have a financial bet with an immunologist. I was telling Doug last night, it's 20% bigger than I've ever bet. I'm not much of a gambler. I'm a little nervous. I mean, how many people go over 20%, you know, over the highest bet they've ever made? So I have this 6-cent bet with an immunologist about what, I've never bet more than a nickel, and even now I'm getting a little twitchy thinking about 6 cents. I have a 6-cent bet with an immunologist who thinks that what's happening with the mice is not immunological. And I tell him he's an idiot. I mean, what else do you need other than this? And he goes, no, I think it's something else, some other mechanism, some other thing, some other. And so we got to test it. But what I'm really doing the research for now is to get that guy's 6 cents, frame it, and lord it over him. But I think that in the case of cancer, the process is giving information, using this only metaphorically and loosely. I think that the cure for cancer is an information. Everybody's got cancer. In the sense that, as far as I'm aware, nobody's ever been found who doesn't have it. And this is found by young people who die in accidents. That's the plot line. That's where you get the database. You got cancer all through you. Don't worry about the stuff. You also got a lot of other crap going through you. Don't worry about that either. You have more non-human cells in you than human cells. That gets me a little twitchy, but yeah, I'm kind of a parasite on myself, you know, which gets me a little twitchy. But you have, as you have other stuff, and you got stuff you want to get rid of, and your body gets rid of it, and here's a cancer cell, get rid of it. That's very different than having a disease, where you get behind it, and it wins. I think that you're giving information to the body. Never trust what I'm saying. I think you're giving information to the body, which is then fixing it itself. I don't think that we're doing anything whatsoever. I think it's the body doing body stuff. Now, it turns out we can transfer this, though, and you're going to see a little cartoon, not today, I don't think, or not in this particular talk, but we can take, as I mentioned at the very beginning, we can take stuff out of a mouse that's been treated and put it into an infected mouse, and that infected mouse, without hocus pocus, will be cured. Biological memory. Proposition 3. There's a dose response, and for this I have experimental evidence, but I have no clinical experience whatsoever. And what I mean by that is, if you've got an upright, you don't say, well, I'd like to play around with taking away dose and see what happens. If you're doing your clinical practice as a vet, I don't think you're going to do that either. So you got 10 dogs with cancer. Well, I think I'll cure these 2 with a lot, but this I'll only do a little. I mean, that's not going to play well with your clients. And so, this is going to be used as evidence against me. I'm really screwed. And so, so clinically, you don't play with dose response questions, but I can do that experimentally in the lab. And so the question will be, what happens if I treat it, for example, what happens if I treat an hour at a time? What happens if I treat a half hour at a time? What happens if I treat 15 minutes at a time? What happens if I treat once a week? What happens if I treat 3 times a week? What happens if I treat 5 times a week? You get the plot. What happens if I treat 7 times a week? We've done all of these permutations trying to find out, is there a minimum amount of treatment, and does it matter, what matters in terms of producing this? How much do we have to keep going? But here's a question in terms of conceptually, how should you think about healing? Is healing a process that starts and has its own inertia, or does it have to be reinforced? And I would ask the same question about everybody in the display booths out there. So they got this, they got the drop, they got so same thing, you got a therapy. Well, if I'm a body in need, and I'm a mouse in need, does healing, which otherwise wouldn't happen, meaning the mice are going to die, if you give it a push, hocus pocus, you give it a push, does it then, like a rock at the top of a hill, start to take on its own inertia? And then it's got its own life. Or do you have to keep chasing it down the hill, pushing it? If I take the drop, do I need a drop tomorrow? Well, yeah, but you've never tested it the way I test. Does healing of all types need constant reinforcement? Or does you hit a phase transition, a change in character, and then nature will take its course whether you're there or not? You can see the permutations and the possibilities. Very few people look into this, particularly clinicians, because clinicians will say, I've actually had a doctor friend of mine, an MD kind of guy, and he's sitting there, and we're there, and he says, I want to learn your techniques so I can fix my patients. I say, yeah, but I don't know this, I don't know this, I don't know this. And this guy, who I call the village idiot, says, who cares? It works. I said, who cares? It works. I care. I don't care about the upright. My question is, what can I learn? And maybe I'm chasing this rock down the hill trying to pretend I'm treating it, and all it does is you need a critical mass to begin. You need to move the rock, and then nature will take its course. You can't play with that in clinical practice. You can't have a cat and say, well, Mr. So-and-so, I'm not going to give it the full treatment because it'll be interesting. That's not going to play clinically. And so you can only do that stuff experimentally. And if we have a clinical case of a person, you treat until they're cured. Did you need to treat until they're cured? I don't know. That's the issue. So this is gobbledygook. Here's the deal. We've done 15 minutes at a time, 30 minutes at a time, 45 minutes, an hour at a time, once a week, 3 times a week, 5 times, blah, 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 and all the permutations. There's only one predictor of the speed of cure. And this is going to come into play later, not this talk. And it's the number of mice in an experiment. The more mice in an experiment, the faster they collectively get better. I suspect this has tremendous clinical implications. And when we first started this 170 years ago, we were doing people, HIPAA laws and all that stuff. You got to pretend you're the only person there and nobody else knows about you. Well, we were all hanging out in the living room, curing everybody. And then people who got cured would just hang around out of curiosity. And so we had, lots of people. Boy, does that work well when you got lots of people. Particularly, I'm going to suggest in my next talk, if they become a bonded group. And I think you folks, not dealing with uprights, have some tremendous opportunities, which I'm going to suggest to you in terms of clinical practice of bonding animals together. And it turns out the most important predictor of collective cure is the number of mice in an experiment. 25 mice will take longer to cure than 75. 5 mice are harder to cure than 10. And the more you have, the faster they collectively get cured. And this will have to do with a phenomenon I'm not going to talk about here, which I'm going to call resonant bonding. So the amount of treatment needed to remit any type of cancer is unknown. There's also an enormous variety of clinical responses. So some people get better very, very fast, and some people take a long time. And we don't know, again, statistically, we call it unexplained variance, which is jargon for, hell, I don't know. So we get in the same cage identical mice, essentially clones, with the same healer and the same food and the same water and the same. One mouse will take 2.5 times as long to be cured as its companion in the cage. Injected with the exact number of cancer cells on the same day. I got nothing. In the uprights, I've had a stage 4 get better in 5 minutes. I've had a stage 4 take months. I got nothing. Unexplained variance, which means. So a speculation that we had was remission rate, is it related to mass or metabolic rate? So we did an entire cancer lab once. They weren't happy. It was actually to study conductivity of tumors. All sorts of animals of various types. We did the whole room. And it turns out the smaller the animal, the faster it got cured. So the mice went, the rats went later, and then. And it worked its way up the food chain. So my question was, is it mass or metabolic rate? And so we started to do 10 mice at a time versus 5 mice at a time, so to equal the speed of another kind of animal. I mean, we've worked this thing through. Turns out it's just number of mice. And unknown, particularly when you have a human factor, is that each dose equal. So here I am, a human treating an animal. Am I doing the same thing on Monday as I am on Wednesday? I mean, is it like a toggle? If I treat for an hour, is it different than treating for 2 times a half hour? Is there a certain amount of treatment that a body can take in, and is that related to the metabolic rate of the size of the organism you're treating? So I can keep going? And you should be sitting there going, hell, I don't know. Just the answers. The answer is very simple. I don't know. And what I'm trying to say is I've looked into this. Nobody knows, but they're making the stuff up. If you're looking for an answer, it's the wrong question. Proposition 4, healing occurs in bursts. And this experimental evidence and clinical experience. I'm going to show you an amazing set of pictures, and I got to tell you, it's amazing. I'm glad you're sitting. Here's the mouse, 22 days subsequent to injection. Tumor's not, or the ulceration's not as big as the last one, but you can see bright-eyed, bushy-tailed, nice coat, the whole thing like that. It's running around a cage, it's having a good time. It's gone through, it's been treated since about day 3, and the tumor grows, the tumor always grows, and the tumor ulcerates, the tumor's ulcerating, and you think, well, this isn't good, this isn't good, this isn't good. I'm going to show you the same mouse 6 days later. That's the biologist pulling back the fur, looking for where was it. So we go from 6 days later, and if you don't pull it back, there's no sign it ever had cancer. And of course, notice the day, it's 35 days subsequent to injection. Remember, the world record's 27. That's not interesting. This mouse is cured. It's going to live 2 years. Once it goes through it gets a blackened area on the tumor, we know the next stage, the next stage, the next stage. 100% predictability, it's good, it's cured. It's just going to take a while before it gets there. Now, you're a person. You're a dog. You're the cat owner. And you come in and you got an ugly looking dog with an ugly looking thing, and you go hocus pocus on this thing, and because you're using my method, you don't actually say hocus pocus, but you're doing my hocus pocus method, and the tumor's getting bigger. Now I'm the owner of the cat. And I say, excuse me, was I not clear? You had a tumor this big and now it's this big. Why am I paying you? Well, just hang in there. Now, the people who have been cured are either the bravest or the dumbest people I've ever met. Because it's not going the way they want it. In the case of mice, they're not brave, they're trapped. But it's the same thing. So you got an upright, you got a dog, and nature is not conforming to your wishes. First, it's got to look ugly, and then there's a sudden phase transition. But if you're going there and I'm the owner of the dog, and the tumor's getting bigger, you going back to that vet? If you're an upright and you go to a hocus pocus guy, hocus pocus, and the tumor gets bigger, you going back to that hocus pocus guy? You're either nuts, no learning curve, brain dead. I don't know how to hierarchically put that. But the people who come to get to full cure are either dumb or brave. You decide. But clinically, let's say the folks here who want to learn how to cure cancer in their animals, it's not a pretty thing. Here's let's go bad, good. Treatment. It is nonlinear. It's not treatment, it gets better, treatment gets better, treatment gets better. You're going to ride a little roller coaster here before the whole thing collapses. And it collapses like a phase transition. So by phase transition means sudden transformation with a little, you take water and make it a little warmer, and make it a little warmer, and make it a little warmer, and suddenly it turns into a gas. That's a phase transition. You got water, it's a liquid, you cool it, and suddenly it's solid. And it's sudden. Healing is sudden. And this suddenness that you just saw in the mouse, that you just saw in the mouse, was sudden, and it was across all the cages. So we're going along various tumor sizes, various ulceration sizes, and you cut 6 days, and it's just astonishing what happened in those 6 days. Now, if I had stopped the experiment, now you can translate that into clinical stuff. If you had stopped the clinical treatment, and so here I treated, I'm making this up now, I treated for 22 days. Not really, but I treated for 22 days, and you're the owner of the mouse. And I'm going, I'm treating, and you're going, well, this doesn't look good. If you had just gone a couple of days more, blam. That makes for a difficult formula because the unexplained variance among the different outcomes of the same lab animal is astonishing. So this mouse, 28 days, and it's clearly at full cure, but its companion, I'm making this up, is 2.5 times as long before it gets to that stage. You come in with Spot the dog, how long is a reasonable question will it take before it's cured? And the answer is, I don't know. When will I start? I don't know. What? I don't know. And that's the true answer. Will Spot be cured? Yes. Not magically. And it may go through a couple of bumps, and it may suddenly look, and it may. So how do you folks, because I don't do clinical stuff, how do you folks who decide you want to fix your animals of cancer, how do you get through that? Your clinical experience is past my pay grade, and I don't know how to give you the answer. Yeah. You met.
We can't help you. It's going to cost you a lot of money. You should be prepared to suffer for 2 years. That's what we tell every single person who comes.
Yeah.
And your dog may die and you're not get better. And it may live like all these dogs on the wall.
Yep.
But that's all we can do. And this is a great technique. And first, I think maybe that by training the owners how to do the procedure.
Yeah.
And making it a group of people who come together either nightly or twice a week or whatever.
Now here's and I completely agree with you. It's harder to do with uprights because they're going to go, well, damn, look at this. I mean, was I unclear what I asked for? You know, you're walking around with this.
But when you do it as a group and you have this in my way, I'm just laughing. I'm not at you.
Oh, it's fine to laugh at me too. I'm okay.
My practice is very small and it's very long.
Yeah.
And so we have a very small waiting room and there's always 3 to 10 people sitting in the waiting room. And the overflow are 4 folding chairs in the parking lot.
Yeah.
And so they're sitting there. What they do while they wait to get in to see us, which is like an hour, even if they're on the appointment, is they go through this socialization period where
Yeah.
says, look how bad my tumor is. No one says, oh, mine look like that. Oh, mine.
That's a beautiful thing.
They do this exchange, which is what you're.
That's a beautiful thing. Yep. I'd be a fan of that. It's harder to do with uprights, and you folks have an advantage. Because here's the deal. Here's what's going to happen. You got Spot. And Spot's got an ugly looking tumor, and I got a freaking out owner. I told you, and they're flopping on the floor and doing all that kind of stuff. Here's what you do to Spot. Hold out your hand. The dog will come over to you, and will come over so long as it needs treatment. I'm going to show you a picture, not today, of mice. And incidentally, everybody who learns my healing method becomes left-handed. So you got to be warned on the way in. There's never been an exception.
And when the dog comes over to your left hand, the owner goes, he hates everybody.
Yep. And the dog will come over to the left hand. So you, because you're not dealing with brain-dead uprights, it's a beautiful thing. All you need to do is offer to the animal. The animal will know how long the treatment is. The animal will need to know how long to treat in a given treatment. The animal will tell you when they're done. Mice do it, dogs do it, cats do it. They do it. And you want to hear some great animal stories, talk to Margaret at the break. Children do it too. And so children will know what to do. They'll know the dose. If you're doing this by formula, it varies. Life is dynamic, folks. You can't reduce it to a little formula. The kids will come over as long as they need it. The dogs will come over as long as they need it. Alzheimer's patients will come over as long as they need it. It turns out we can fix Alzheimer's pretty quick. We can't do age-related dementia. If you're just decomposing and your brain is shrinking and you're shrinking, you're at the end stage. If you've got crap on the brain, we can get rid of the crap.
Why left-hand?
Left-handed people are inherently superior. Did I joke? No, I'm speaking empirically, you know, but I don't know. I really don't. I've done EEG stuff. I've done functional MRI stuff. I've thought, you know, well, therefore it's located in the right hemisphere and all that stuff. I thought I had isolated in functional MRIs a right parietal lobe I mean, nothing. I got nothing.
And it's only for healing. It's not like you're gonna learn to write.
Yeah. Margaret is still handicapped. She uses this. I can't remember why, but it's, you know, it's something unnatural on her part. Yeah. You can speak to her and maybe you can fix her. But when she heals left-handed, if you're doing this technique. We don't know why. In any event, Doug's going to yell at me in a second. So I'm going to not do anything. Okay, so we have sudden bursts of healing, and in case of people, it's the same thing. And we find interesting correlations found when we run gizmos. Random event generators, geomagnetic probes, and things along those lines. So this is gobbledygook for stuff that we do. You saw when the picture of me treating in Indiana, we were running geomagnetic probes, we're running random number generators, we're doing this and that. We also, and this is really, really interesting, so imagine we've got an animal room here, and think of the picture you just saw, and we got cages, we're doing all that stuff. Next door to that is another animal room, next door to that is another animal room, and then out here is the hallways in the animal room or the animal building and all that kind of stuff. We've got probes in various orientations in all the rooms, and it turns out the physical space of the room changes in bursts. So if you're running, let me just do this quick. This is noise down here. This is like a noise output. And up here, you can see the difference between this and this. So this is what's supposed to happen, and it's what's happening in this room right now. But if we do hocus pocus, and we've got cancerous mice in the room, then between 60 and 120 seconds, we have as if it's a reduction in entropy in the room. Now this happens simultaneously in all the animal rooms we're in and no place else on campus. And for burst, random number generators don't generate random numbers. So it's like an electronic coin flipper doing heads and tails. Well, it's a chaotic chance effect, but it starts to not become chance, as if you're inserting information into a system and it's a reduction in entropy in the room. This only occurs when the mice have cancer. Once they're cured, these things don't occur. Random numbers generate random numbers. Random event or geomagnetic probes just give a chaotic output. But if there's a need expressed in the room, something happens in the physical space. Later, way in the afternoon, for those of you who are masochists, later, way in the afternoon, when we talk about trying to record this stuff, it turns out we actually change the internal stuff in an incubator by running experiments inside of it. And that stuff lingers in the incubator. When we change a room, it has a lingering effect. The physical space itself is altered. There's some interesting correlations to this stuff, and I don't know. That's going to be my answer. Proposition 5, I'm getting flagged. Some types of energy healing may not be compatible with some types of conventional treatment. And for this, I have clinical experience. I don't have any experiments. It would be really simple to do this as an experiment. It just hasn't been done yet. It's on the scroll. So if we radiate a mouse and then inject it with cancer and then do hocus pocus, if we treat a mouse and then radiate it, you can see the permutations. But we haven't done it yet because I got hundreds of experiments to do. I haven't done yet. Just time, money, labs, all that kind of stuff. But in terms of clinical experience, you can imagine uprights, and my clinical experience is not going to be about dogs and cats and such. We're talking about uprights. In the uprights, there's a tendency to want to throw the kitchen sink. And certainly from a psychological point of view, that's reasonable. You know, give me whatever you got. You got vitamins, I'll take them. You got radiation, I'll take it. You got hocus pocus, I'll try anything. I got to get rid of this stupid thing. So you can understand it, but it turns out that all the clinical failures have been associated with combining it with conventional treatments that kill. And I've got here speculation, maybe healing and killing are incompatible. And I don't have experimental verification of this. So this is to be determined. These are the funders for what you've seen so far. I got other funders for other parts of the program. I'll give you that as a... I like this. and thank you, and now we'll take questions for a couple of minutes. Yes?